A pleasant day to everyone. I am Maria Jessica C. Bacolo, and I will be presenting a research entitled Cecid Fly Defect Detection in Mangoes using Object Detection Frameworks, along with my co-authors, Dr. Conrado Ruiz and Dr. Oya Aran. The Carabao mango is considered as a major agricultural produce in the Philippines due to its high export value, making the Philippines as one of the top 10 exporters of mangoes worldwide. However, in the recent years, the production yield has experienced significant decline due to Cecid fly infestation. The Cecid fly defect produces a circular blemish on the mango surface that affects its quality, quantity, and market value. The first major infestation was recorded in 2013, which affected up to 90% of the mango production yield. At present, many plantations succumbed to these attacks as there are no insecticides are yet proven to eliminate the Cecid flies. Experts and government agencies advise large and small-scale farmers to practice efficient pest management such as field sanitation, bagging, pruning, and chemical sprays. However, due to the lack of training and information dissemination, small-scale farmers opt to use fogging and chemical sprays for all types of defects. Image processing and machine learning algorithms have been previously used to automatically detect surface defects in fruits and leaves, as shown in the following. In addition to this, there were also studies which focused on the defects of mango fruits, flowers, and leaves, such as the following. Based from our review of related works, the following observations were made. 1. The dataset and model for the detection of Cecid fly defect were not yet studied. And 2. The trained classifiers were trained and tested on images that contain one exclusive defect without localization. Based from the ocular analysis of the images gathered, the defects in mangoes may contain one or more surface blemish, and some belonging to multiple types of connected defects. In line with this, our study aims to detect Cecid fly defects using images with multiple blemishes with the use of object detection frameworks. A total of 1,329 surface defects were collected from 318 Carabao mango pictures to create our dataset. The images were taken in both natural and artificial lighting with a whiteboard as a backdrop, and each image contains exactly one mango object. 10% or 31 images from the dataset containing 199 blemishes were used for testing. The images were pre-processed to perform color correction and enhance the features of the input images due to the variation caused by the non-uniform light sources. These images were processed with automatic white balance, gamma correction, and image sharpening. Our proposed approach replaces the predefined region search algorithm with a segmentation technique to extract the region proposals. To get the binary masks, we used adaptive binarization and thresholding for the hue and value channels. After converting the RGB image to grayscale, adaptive binarization was used to compute a threshold for each pixel based on the local mean intensity in the pixel's vicinity. The resulting masks were aggregated and cleaned using morphological operators. Each of the blemishes are isolated by computing the crop parameters. These parameters were also used as the values needed for the bounding box of these blemishes. 5 object detection frameworks were compared to identify the defects found in Carabao mangoes. 3 of these frameworks are state-of-the-art methods, namely the region-based CNN, the fast region-based convolutional neural network, and the faster region-based convolutional networks shown in these slides. In addition to these frameworks, the modified versions of region-based and fast region-based CNNs are proposed. In these modified versions, we replace the region search algorithms with our proposed image segmentation technique. The following slides contains the specifications for the convolutional layers used to extract the feature vectors for the various frameworks. The frameworks presented in the experiments were trained using 3 different CNN depths, including a 3-layered, 5-layered convolutional layers, and a pre-trained VGG16. The predefined weights in VGG16 were used to train the 3- and 5-layered architectures from scratch. The presented graphs illustrate the performance of the models with respect to its average precision in 2 IOU thresholds and inference time. The blue bubble represents RCNN, red represents modified RCNN, yellow for FRCNN, green for the modified FRCNN, and orange for faster RCNN. Upon testing the trained models, the results show that the proposed models, MRCNN and modified FRCNN, achieved higher average precision and faster detection time for both IOU thresholds than their counterparts, RCNN and FRCNN respectively. Faster RCNN had the highest average precision of 0.901 at the 0.5 IOU threshold, while the modified FRCNN had the highest average precision of 0.723 at 0.75 IOU among all 5 models. Even though the average precision of the MRCNN, FRCNN, and MFRCNN is comparable to that of the faster RCNN, the latter was able to complete the detection faster. However, when the IOU is set to a higher threshold, MFRCNN outperforms all other frameworks as seen in the confusion matrices. Here are some sample detections generated by the trained models. In a test image containing 1 defect, it can be noticed that all of them were able to classify and locate the defect with minimal difference on the localization. It is also interesting to note that our model was able to locate smaller blemishes which were not annotated by the human classifier. For images with smaller surface defects, we can notice that the model trained with modified fast RCNN was able to accurately detect most of the surface defects in comparison with the other models. Based on our results, our proposed approach worked best with these types of images. Another observation can be made in this test image. The blemishes found at the top left of the mango was annotated in 1 bounding box due to its proximity. We can see here that our modified approach generated separate detections for such cases, which may be useful for multiple types of defects. We also observed that all models had difficulty in locating connected blemishes as shown in this slide. The results of the experiments presented in this study show that object detection frameworks can be used to locate and classify fine-grained blemishes on mango fruits. We also conclude that the modifications done to extract the region proposals further improve the performance of the models at a higher IOU threshold. Furthermore, different lighting conditions, size variations, blemish proximity, and blemish connectivity can all contribute to difficulty in detection. We plan to collect and annotate more images from multiple defect classes in the future, as well as train a multi-class defect detection model. Additional tuning of the FRCNN, faster RCNN may be done, as well as the improvement of feature extraction using deeper CNN architectures. The development of a real-time and accurate defect detection model could be used as a part of an automated framework. This is the end of our presentation. Thank you very much for listening.